Okay, today I want us to look at industrial fermenters. This is the section that we want to look at how the industrial fermenters can be used in fermentation. Now, when we talk about a fermenter, the term fermenter is from the word ferment and the term fermentation. Now, when we talk of a fermenter, this one is usually a large vessel. That's how we can describe it. And this one is used to grow microorganisms. And these ones, the microorganisms, are now used in the process of fermentation. Now, industrial fermenters, the ones that have been mentioned here, actually these are large tanks, and these tanks can be even up to around 200 decimeter cubic in size, knowing very well that 1 dm cubed is equivalent to 1 liter, meaning this one is equivalent to 200,000 liters. That is a very large quantity, and meaning that this one is the liquid culture that they are able to do what? To carry. Now, for fermenters to perform their function to carry out their function properly of fermentation using that liquid culture, there are environmental conditions that need to be controlled so that you can obtain high yields of the products. So I can say, to obtain high yields of the product, the following environmental conditions must be controlled. And when we talk about environmental conditions, we are looking at things like temperature as one of them, knowing very well that temperature affects enzyme controlled reactions. Now you realize the enzymes are affected by temperatures beyond the optimum and temperatures below the optimum. Then two, oxygen also needs to be controlled, and this one depends if it's the aerobic microorganisms you are dealing with, or that is anaerobic microorganisms or aerobic microorganisms. Now, when you talk about anaerobic microorganisms, these ones, they respire in the absence of oxygen. But when you talk about aerobic, these ones, they respire in the presence of oxygen. Now, another factor that needs to be controlled is the carbon dioxide levels, as carbon dioxide concentration. Now, you know very well that when carbon dioxide levels increase, these ones, they can even, they reduce the pH. So when the carbon dioxide levels increase, the pH decreases. And you know, also enzymes are pH specific. Enzymes are pH specific, and therefore, if the carbon dioxide levels are not controlled, they can equally affect the functioning of the microorganisms because their metabolic reactions are controlled by enzymes. So they need also to be controlled. That the carbon dioxide levels needs to be controlled. Then there is what we refer as the pH. As I've said, the enzyme controlled reactions are pH specific. They are pH specific. Then we have nutrients. Remember, nutrients are in this case substrates, especially, for example, let's talk about glucose. These are respiratory substrate for most of the organisms, including now these microorganisms, which require glucose to break it down to produce, for example, ethanol, if you are making beer, for example, and carbon dioxide. Now, I want us to look at the basic structure of a fermenter, and the diagram you can see is there is the basic structure of a fermenter. Now, we have the motor here. This one is to rotate these stirring paddles here so that they are able to mix the contents, and this keeps the microorganisms suspended. Now, why do we need to stir the mixture? These are the 2 points that you need to note. One, it keeps the microorganisms in suspension in the liquid so that they get more exposure to the nutrients. That is the first point. Then two, it is to keep the temperature even throughout the vessel. You can use same, or you can say even distribution of the heat throughout the vessel. Now you realize that some fermenters, they use jets of air to mix the contents instead of using these stirring paddles. You know, when air passes through a liquid, those bubbles, they mix the liquid. Then we have the steam inlet. This one is to bring in the steam to keep this vessel, especially when we are carrying out sterilization. This is the steam outlet to remove the steam once it comes in during the sterilization, especially after a fermentation process. You need to stir this, to clean this. Then at the beginning of this fermentation process, you realize that all the nutrients are fed through these inlet pipes, and they contain valves so that they can control the substances that come in and out of this. Now, there is what we refer as the temperature probes here. Now, these temperature probes and pH probes, they are used to monitor. Actually, they are used in monitoring the temperature levels. So you can say they are used for monitoring the temperature levels and the pH levels. Now, these temperature levels, when they go high, they are actually returned to normal through different ways. Now, the main way is actually using a water jacket that is surrounding this one that has been labeled here. This water jacket is actually used to regulate, to bring the temperatures to normal, to prevent the contents from overheating. What has been mentioned here actually, when we talk about the fermentation produces heat, because remember when we said when you look at glucose, when it undergoes fermentation, it will produce ethanol as one of the products, like in this case. Then it will produce carbon dioxide and also energy. Now this energy is the one that comes out in form of heat, so that now that heat that is produced, for you to prevent the contents from overheating, remember temperature affects enzyme controlled reactions. So to prevent the contents from overheating, the fermenter is usually surrounded by this water jacket which has been mentioned here, so that now as the water circulates, the cold water circulates around this, it keeps the temperature at a constant level and the level that is best for the growth of the microorganisms. This is the optimum temperature for the growth of microorganisms. Then we have the pH contents. This one can be regulated by adding either the acid or the alkali. The acid is to reduce the pH. This one is to reduce pH. And the alkali is to increase pH, knowing very well that when you add an alkali, you increase the pH, and when you add an acid, you reduce the pH. Now, there is a air inlet. Now you realize that if organisms are aerobic, those that respire aerobically, they will need oxygen. So for oxygen to come in, we need air inlet. So this one, the air inlet contains a filter, so that when it comes in, there are no microorganisms coming through. That is maybe the air that is coming in should not be carrying microorganisms. So that is why they are saying here that if the microorganisms respire aerobically, filtered air is applied through this inlet. The air is filtered to prevent contamination, meaning to prevent entry of microorganisms into the vessel, that is into the fermenter. Now, so if, for example, you are making a fermenter, you need to make sure that it is made up of stainless steel. The reason behind that is because these microorganisms produce some of the products that are corrosive. For example, they may be producing, some may be producing, for example, lactic acid. For example, when you look at those that are used to make yogurt, they produce lactic acid. That's a lactic acid bacteria like the streptococcus and lactobacillus. they produce lactic acid, which is corrosive. So to make a fermenter, you need stainless steel or alloys. They refer them as special alloys, which are not easily corroded by the presence of these products or materials that are produced by the microorganisms. Now, I want us to look at the next thing about the effect of using unsterilized fermenter and nutrients. Now, let's say you are carrying out your process of fermentation and you need uh, you need to keep the fermenter sterilized and the nutrients sterilized. Now, we want to see what are the likelihood of the effects of using a sterilized fermenter and nutrients. Now, first, I want you to note that when you are finished making your you have finished carrying the fermentation process, meaning when you say fermentation is finished, what happens is that the products are collected. Now, these products are collected from where? These products are collected from the outlet pipe. Now, once collected, the fermenter then needs to be filled with the new nutrients. So we must have new filling. Now, before new filling, the fermenter is cleaned and sterilized. Now, during the cleaning of this fermenter, there are methods of cleaning it. Now, one of the methods is to use hot water, or we can say steam, which is under high pressure. Then two, you can use a 70% alcohol disinfectant. Now, I want you to go with the first method of disinfecting, because this one, even if it remains in the fermenter, it does not destroy the microorganisms required for the process of fermentation. The microorganisms required in fermentation. But the second one, if the ethanol remains in the fermenter and you bring in the new microorganisms that you want to refill the fermenter with, they are likely to be destroyed by the alcohol disinfectant that if it is not well cleaned or removed from the fermenter. Now, why do we need a fermenter that is sterilized and nutrients that are sterilized? What if you use those unsterilized fermenter, let's say unsterilized nutrients and fermenter? Now, one thing is that the microorganisms, if they manage to get into the vessel, so the microorganisms may compete with those useful, there are those ones that are useful for this process, the one that you require for the process of fermentation. Now, so we can say the microorganisms may compete with the organisms in the culture. So with the organisms in the culture, reducing the yields. That is one thing that is likely to happen, meaning they are likely to compete with them for the food nutrients, for oxygen, all that. So the resources they will compete for is, let's say, oxygen. We can talk of nutrients themselves. Yeah. So this one will affect the yield of the products. Actually, it will reduce the yield of the products. Then two, the products that formed from this process, the products may be contaminated, may end up being contaminated by the waste products that come from those microorganisms, by the waste products from those microorganisms. Now, how do we prevent contamination of unwanted microorganisms? That's what I want us to look at, ways of preventing contamination within the fermenter. One way is actually filtering the air. Filtering the air, and this air is coming from the air inlet. So you make sure that there are filters there to filter that air. Then two is by the use of sterile nutrients. Use of sterile nutrients. This one is to prevent the contamination of the microorganisms that are within that fermenter, the one that are you require, and also to prevent the contamination of the products. Then three is sterilizing the fermenter by using what we refer as aseptic techniques. So is sterilizing the fermenter using aseptic techniques like the ones I mentioned in terms of using hot steam and high pressure or using disinfectants. Then another thing is that apart from ways and preventing contamination, I want you to learn that some of the fermenters are highly automated in that they are able to monitor the temperatures, the pH, the oxygen, the carbon dioxide levels, and also to provide the optimum environmental conditions for the better growth of the microorganisms.